Thank you so much, Paula. Since we're a couple minutes over time, I think I'm going to ask if you could answer some questions in the chat, and if people could ask their questions in the chat. And now I'd like to introduce Vitor Borges from the National Institute of Health in Portugal, who will be talking about the first monkeypox virus genome that was sequenced this year.
Thank you. Can you see the full screen?
Yes, looks great.
Okay, thank you so much. It is hello everyone. It is a great pleasure to be here today, and I really need to thank to the organization team for giving me this opportunity. So I work at Genomics and Bioinformatics Unit of the National Institute of Health in Portugal, and we are running the genomic surveillance of SARS-CoV-2 and now monkeypox. And today I will just talk about our contribution in the first stage of the outbreak, this large outbreak, with the first phylogenomic characterization and signs of microevolution. So I would like to start with this very timely revision, just published 3 months before the detection of this large outbreak, where the authors showed highlighted an escalation of monkeypox cases, particularly among young adults. They also raised the awareness about the appearance of outbreaks beyond Africa and stressed the need for increased surveillance and detection of cases, as of course, as you know, for prevention and control of dissemination of this virus. So we all know that three months later, we first faced a multi-country outbreak since the first case has been reported on UK on 7 May. Cases have been reported in multiple countries that do not usually have monkeypox virus in animals or human populations, and we have now more than 60,000 cases spread among more than 100 countries. I must highlight that 12 countries, including Portugal, account for 90% of the reported cases. So I will not go into details on the epidemiological and clinical characteristics of this outbreak. It's not the purpose of this symposium, but I do recommend this systematic revision that has been just published last week, and they provide a very good overview of the epidemiological and clinical characteristics of thousands of cases across 41 countries in Europe. So in Portugal, all suspected samples are sent to our institute for diagnosis. The first confirmed cases were on 17 May 2022, and we had more 20 cases on next day. So our team was called to do the sequencing. At the time, in that day, we selected 2 samples with a good viral load, and the laboratory had performed a regular DNA extraction, and we didn't perform any human depletion, and we rapidly prepare a library and put it in a mini ion and let run overnight. So in next day, we were able to obtain 0.5% of monkeypox reach. So after a brief draft assembly, we selected a closed reference genome to be used for the analysis. The reference-based analysis was performed in our platform, InsaFlu, which is a free online open tool that we are using for routine genomic surveillance of influenza, SARS-CoV-2, and now monkeypox. And at that time, we inspected carefully all the mutations so that we could provide a reliable or robust phylogenetic placement of this 20,002 outbreak virus. So we succeed in releasing the first draft genome sequencing of this associated with this outbreak. The sequence has been released at that night on virologic.org, and which triggered a very nice discussion that I recommend, a very good discussion about the regions and the evolution of the virus. 4 days later, we are able to release more 9 sequences and provide the first insights in the genetic diversity and microevolution of this outbreak-causing virus. So this culminated in this publication about the beginning of the outbreak, whose main results I will try to summarize in the next slides. So this shotgun metagenomics allow us the reconstruction of the first outbreak sequences and allow us to confirm that this virus belongs to the clade 2B, which is associated with less fatality rate when comparing with clade 1. Not unexpectedly, all outbreak sequences clustered together, suggesting that the ongoing outbreak has a single origin. But surprisingly, the outbreak sequences segregated in a considerable divergent phylogenetic branch, diverging from a cluster of cases from 2018 and 19 that were linked to an endemic country, in this case Nigeria, where an important outbreak took place in 2017 and 18. In fact, we found out that outbreak sequences have more or less 50 SNPs from those viruses from 2018 and 19. And this was surprising because it would represent probably 6 to 12-fold more than we would expect, considering the previous estimates of the substitution rate for this kind of virus, which is about 1 to 2 substitutions per genome per year. So the hypothesis that these outbreak virus may be undergoing a continuous accelerated evolution. So the hypothesis was that the emergence of this outbreak resulted at some point in the time most likely from importations from an endemic country, with this 2022 virus potentially representing the continuous circulation and evolution of the virus that also caused those cases in Nigeria in 2017 and 18, and also travel-associated cases that have been detected since then. I'm not highlighting here sequences reported in 2021 because it will, of course, be the focus of our next speaker, Professor Crystal, and for sure will help understanding this scenario. So, as I said, this triggered a very good discussion in virology.org, and I must say that Andrew Rambo from the University of Edinburgh brilliantly launched a discussion because they found a strong mutational bias in this large divergent branch associated with this outbreak, where the most mutations have this pattern, these two dinucleotide replacements that you can see in red. So they suggested that this could be due to the action of ApoBac3 enzymes in viral genome editing, and this could be the reason behind the accelerated viral evolution. So for those that are not familiar with this process, the expression of these host enzymes can increase in response to the viral infection, and these enzymes will try to introduce mutations in the viral genome, trying to destroy the virus. But sometimes this simply led to the fixation of mutations, and the virus survives, and this give it the opportunity to evolve and to generate viral variants. So in our work, we inspect for these ApoBac3 signature mutations, and we found them at 3 levels. As I said, in level 1, in the branch leading to the segregation of the outbreak causing virus. We also found an excess of this type of mutations during the diversification that occurs throughout the outbreak transmission chain, and also for the few samples that we had at that time with a very good coverage, we also inspected the viral subpopulation diversity at interpatient level, and again, we found an excess of mutations with this signature. So this supports the idea, the trigger initially by Andrew Rambo, that we are seeing a potential continuous action of this ApoBac3 in the evolution of monkeypox. Of course, we also looked for the sequence that we analyzed at that time. We finally inspect the proteins that were being targeted by mutations, and we found some potential signs of human adaptation. Indeed, again, in those 3 levels, we found, for instance, three amino acid changes in a particular protein, and immunogenic surface glycoprotein B22. With serologic studies have previously indicated that this protein could be an important antibody target. When you look at that few mutations occurring during the outbreak, we also found again same pattern, and again also one particular mutation in this protein. And we also found a subcluster with a frameshift deletion that could be important because gene loss was also previously associated with some human transmission. And at level 3, when we found when we inspect the mutations at the interpatient level, we again found a few mutated proteins that are known to interact with the human system. So in these host virus arm race that take place during viral infection, we are seeing the evolution of the virus. We don't know, of course, if these proteins play a role in human adaptation, but for sure they can be good targets for future functional studies. So in summary, we try to contribute to give the first insights in the evolutionary trajectory of this outbreak strange. The observed accelerated evolution may suggest that viral genome sequencing may provide sufficient resolution to track the transmission dynamics. And we think that our strategy, the strategy that we adopt with real-time data sharing, together with other studies, of course, that recently came out, may help guiding novel control measures and, of course, subsequent research directions. Just to end, I just want to highlight that it was an honor to be part of the first group, a small group of scientists that initially proposed a novel non-discriminatory nomenclature for monkeypox clades, trying to remove the geography from the names, which is against best practices, and I think is something that will happen for other pathogens, for other viruses, trying to remove the geography from the clades and lineage classification. I'm also trying to contribute in a small group, in a group that is trying to designate new lineages. We'll have a very good talk today about the lineage by Emma and Cornelius, but my little contribution has been to using a tool that we have recently developed, Report Tree. And with this tool, what I usually do is to cut the tree, to get the clusters, and then have some very nice reports about about these clusters, how many regions they involve, how many countries, the time span of these clusters, and this may help to identify some growing lineages that can be candidates for designation. So finally, of course, sequencing is ongoing. We have already sequenced more than 300 sequences, but of course, with the recent development of multiplex PCR empirical schemas, we expect that the activities, genome sequencing activities for monkeypox will scale up at worldwide level, and today we'll have the opportunity also to hear about these new approaches. So finally, I just want to thank again to the organization for the invitation, to all people from our institute that have been involved in this outbreak investigation, and also other collaborators from the public health authorities, and of course to Andrew Rambo's team to raise this very good discussion about ApoVec3 genome editing, and more recently the team from Chantal Vogels for providing us primers for testing this amplicon-based sequencing. Thank you so much, and I will be open for questions.
Thank you so much, Vitor. That was a very interesting talk, and there's quite a few interesting questions in the chat. If you wouldn't mind answering them over there, because I think we are just at time. Fantastic.
Okay.